Thank you, Chris. Thank you, Liana. And thank you, Giovanni, for inviting me to present at this webinar. And I thank you all for connecting. Before we start, I have to disclose my interest. These are only interest at this time, but I really hope that they will become financial very soon. So my area of research, as you know, are studying lipid metabolism and function in pathogenic fungi. We are mainly working on a pathogenic fungus called Cryptococcus neoformans, but recently we are also working on Candida albicans and Aspergillus fumigatus. And on this microorganism, we are studying fungal pathogenesis, and we are also interested in developing a fungal vaccine. Another area of research are sphingolipids, and mainly on fungal sphingolipids, particularly phytoceramide, glucosylceramide, inositol sphingolipids. Although in recent years, we have also been working on understanding the role of host sphingolipid, particularly sphingomyelin synthase and sphingosine kinase, as you can see today, and how these host sphingolipids are controlling the infection, particularly fungal infection. And the reason why we are to work on these pathways because there are several fungal-specific inhibitors that we are trying to develop in the sphingolipid pathway. And here we highlight some of these. Most of these are targeting fungal and mammalian pathway, but some of these, particularly the aureobascidin, the carfenfungin, and also a new compounds that we developed in the acyl hydrazone that block the synthesis of fungal glucosylceramide are particularly interested to us. And indeed, another area of research in my laboratory is the research development of new antifungal, and these are my interests with microarray technology. And here we are working particularly on the acyl hydrazone, which is a new class of antifungal. And we are also working on the ceramide 1, the CER1 fungal-specific inhibitors, and also on another target, which is a steroglucoside 1, which is also fungal-specific inhibitors, and because STJ1 is not found in mammalian cells. But today's talk is about the mammalian cells and is about the immunity against the Cryptococcus and how this immunity is controlled by a compound called FTY720 or FingoLeader. And so before we start, let me summarize the pathogenesis of Cryptococcus. We are basically all infected by Cryptococcus, either by primary infection or by secondary infection. We nail the spore from the environment because this fungus is present mainly in trees and animals, particularly birds. And upon infection, the spore will be deposited in the alveolar space and phagocytized by macrophages or other immunocells. And if you are immunocompetent, you are basically protected against the infection. But there are two models of this pathogenesis of cryptococcosis. A model in which we are all infected during the early stage of our life, during the childhood, and we resolve the primary infection with the granuloma, and only upon immunosuppression, we reactivate the infection from the lung, and upon this reactivation, the fungus can disseminate to other organs, and Cryptococcus like particularly the brain for some reason, and upon infection of the brain, you develop meningoencephalitis. But there is another model of infection in which we are continuously exposed to the fungus. And upon this exposed, we can only be infected only when we are immunocompromised. And if we are immunocompromised, we disseminate to the central nervous system, and therefore you develop a meningoencephalitis. Once you develop a meningoencephalitis, there are holes created on the brain tissue, and it's extremely difficult to treat the infection once you have this kind of picture. Now, both models are probably right, and that's because there are different species of Cryptococcus, and some species may prefer a reactivation model, and some species may prefer a primary infection model. There are studies that has been done by Pasteur Institute in which they isolated 20 years apart a fungal strain that was causing Cryptococcus meningitis from the very same patient in which 20 years earlier got the same identical strain. Considering that there is a lot of genetic diversity in the environment, it's very unlikely that that person got exposed to the very same strain 20 years apart. But again, the primary infection model is still present, particularly in patients that never been exposed to crypto or never had antibody against Cryptococcus. Upon the infection, the crypto comes down in the alveolar space, and once reach the brain, there are a series of events that take event, and these are involving immune cells and more lymphoid tissue. But upon this primary infection, there is a developing of a lung granuloma. This is absolutely important because the granuloma is characterized by a ring of multinucleate cells forming macrophages and a necrotic tissue which is present inside the granuloma. The granuloma is surrounded by a ring of fibroblasts and collagen. And this is very, very similar to the granuloma that is developed by another bacteria called Mycobacterium tuberculosis. The main difference between the two is that the Mycobacterium tuberculosis got calcified, and therefore you can really see it when you do an X-ray, whereas the Cryptococcus does not get calcified, and therefore you don't see it on an X-ray. It has just a collagen deposition, but if you touch it, it's very hard to the touch. It's very, very strong. And the mice model that we have to understand the granuloma just was recently discovered. We have a cryptococcal mutant called delta GCS1, which I will tell you soon what is this mutant. But upon infecting mice with this mutant, we have a containment in the lung granuloma. We have a containment in the lung, and the lung develop a classical granuloma. Whereas if you infect the mice with a very high virulent strain, you have a host fatality and the mice are dying. Whether if you're infecting the mice with a totally virulent cryptococcal strain, the mice are clearing the lung. Somehow there is this mutant, this cryptococcal mutant, that are now is enabling us to study the pathogenesis of this granuloma in mice. And this was discovered about 10 years ago by PhD students in my laboratory. And the mutant consists in the lack of fungal glucosylceramide. What these PhD students did, they delete the gene called glucosylceramide synthase 1. And upon deletion of this glucosylceramide synthase 1, there is no production of glucosylceramide, whereas all the other lipids are basically untouchable. They are basically the same level, including all the IPCs. And if you take this mutant into the animal are all alive. But interestingly, the animals are alive, but they are containing Cryptococcus in the lung environment and are very low level, around 10 to the 4 cells per lung. And if we look at the histopathology of the lung, you will see that the lung is characterized by, as I illustrated before, a classical ring of forming macrophages. There is a necrotic tissue which is inside the macrophages. All these blue things that you see are cryptococcus cell. This is a Movat staining. It's different than any other staining, and it highlights the polysaccharides in blue. And this structure is surrounded by the collagen deposition and by a strong inflammatory response. And this structure is about 1 millimeter in diameter, between 0.5 and 1 millimeter in diameter. And they are surrounded by normal lung, as you can see right here. Now, these cells that you see inside here are alive. These are not dead cells. Now, in contrast, and this is observed about between 30 and 90 days after the infection of this mutant. Now, if you take the wild type Cryptococcus, you don't see this structure. You just, the entire lung is totally destroyed by fungus. And this picture is taken at 20 days, but if you wait a little longer, it's just the mouse are dying because the fungus eventually disseminates to the brain. The characteristic of this model right here is that the Cryptococcus is maintaining the lung and without any dissemination to any other organ. So it is clearly a very nice model for studying the infection that I told you before about the reactivation model. This is more a primary infection model. And here I show you the comparison between the mouse granuloma and the human lung granuloma. You can see that this is a MOVAT staining, this is an H&E staining of this mouse granuloma, and this is an H&E staining of a human granuloma. As you can see, they are very similar, if not identical. Now, a few years ago, we showed that the sphingosine kinase 1 is important for the proper granuloma structure and organization of fungal containment. This was done by a PhD student in my laboratory, and he showed that if you are infecting a mouse in which SK1 has been deleted, you can see that the granuloma is totally different than the wild-type mouse. Here, somehow, the MOVA staining, the color, the Cryptococcus in greenish instead of bluish, but it's very similar. And as you can see that you don't see this structure in the SK1 mutant. But what was most interesting about this model is that none of these mice, SK1 mutant mice, was able to contain crypto in the lung. Indeed, all of them, some more, some less, they have a Cryptococcus in the brain, suggesting that this structure here is not able, when SK1 is deleted, the granuloma is not well formed somehow, and therefore now crypto escape the lung and disseminate in any other organ. Even though all these mice after 100 days of infection were all alive, still they have crypto in the brain, suggesting that eventually they may die. Well, you all know that SK1 is phosphorylating sphingosine to sphingosine 1-phosphate. There are two sphingosine kinase, one is SK1, SK2. SK1 is particularly abundant in the lung, whereas SK2 is particularly present in the liver. And here I show you the role. I show you the focuses within those K1 and SK2, and because of this, we also study what is the effect of S1P upon CN infection. And here I'll show you only one slide. This is important because it highlights that upon infection with Cryptococcus, clearly there is a stimulation in the lung and the hyperproduction of S1P. This S1P was detected secreted in the bronchoalveolar lavage. And as you can see over here, upon infection with, particularly with our mutant, there is a strong activation or secretion of S1P in the lung environment. And whereas in absence, obviously, of SK1, there is no production of S1P in the bronchoalveolar lavage. What was interesting to us that this effect is also present in vitro without any other immunocells, but only with the MHS, which is a cell line for alveolar macrophages. And you can see that if you put the MHS and the Cryptococcus together, and here we have a Cryptococcus wild type and delta GCS1, even particularly in presence of delta GCS1, you have an increase of sphingosine 1-phosphate. And again, this was measured in the medium secreted. Whereas, in absence of MHS, crypto normally does not produce sphingosine 1-phosphate because it produces mainly phytosphingosine 1-phosphate and not sphingosine 1-phosphate. But basically suggests that there is a stimulation by the fungus to the cell and to provide provide the sphingosine production of sphingosine 1-phosphate. Because of this interest that we had on sphingosine and Cryptococcus, we were contacted by Novartis a few years ago. because Novartis was detecting some cases of cryptococcosis in patients taking fingolimod. Now, FTY720, as you know, is a drug that is in use for multiple sclerosis, is targeting a certain sphingosine-1 phosphate receptor. There are 5 sphingosine-1 phosphate receptor, and Gilead and fingolimod was one of the first approved oral in the United States, FDA-approved treatment for remitting multiple sclerosis. And there are, as of February 28, 2021, a year ago, we had approximately 60 cases of cryptococcal meningitis in 299 600 patients treated with FTY720. This corresponded to about 778 and 900 patient year exposure. These combine phase 3 and phase 4 phase 4 is post-marketing. Both phases are contained here. Most of the cases of Cryptococcus were in the United States, followed by Australia and Japan, and 13 cases were fatal. So therefore, there is a concern about an important aspect of this treatment. And so because of this, we were contacted by Novartis. And the work was assigned to Ariel Bryan and Karen Yu. Ariel is now a project manager at the Ingenious Targeting Laboratory in Long Island, whereas Karen Yu is a future MD PhD program. She just has been accepted in the MD PhD program at Case Western in Cleveland. And so there are several S1P receptors, as I told you before, 1 to 5, and the characteristic is that they are present almost in any cell, but certain immunocells, they have a different sphingosine-1 phosphate receptor. For instance, S1PR1, 3, and 4 are almost present in every cell, whereas S1PR2 and 5 are selectively present in certain type of cells. So there are several reviews I'm citing here, but there are about 1,000 reviews about S1P receptor published online, and some of these are focusing on compound that target S1P receptor. And the fingolimod, in order to bind to this receptor, needs to be first phosphorylated. And this is a characteristic of this drug. Whereas other 2 compounds, siponimod and AUY954, do not need to be phosphorylated. They can bind directly to the receptor. And as you know, when they bind to the receptor is internalized, and therefore is not available for S1P to bind to the receptor, and therefore it functions as a functional antagonist of S1P. It works against the multiple sclerosis because it causes a profound lymphopenia, and by preventing the egress of T cells from the lymph node. And this decreases dramatically the symptomatology of the multiple sclerosis. I cite here a few review papers about this issue. I really like the paper, particularly by the Drug Discovery Today, in which highlights how the second generation of immunotherapeutics in multiple sclerosis, and discuss how these new drugs, the pros and cons of these new drugs compared to the old drugs, and whether or not we should totally disregard all drugs. But this obviously it's for a nice reading. But anyhow, so we were provided by Novartis with fingolimod and siponimod, and we test these drugs in our animal model. And we use our primary infection model and our reactivation model. And the experiment we did is we infect mice with our Cryptococcus mutant, and then we let the macrophage, we let the granuloma to develop, and at day 0, after 30 days, we come with our drugs. And as you can see, only treatment with FTY720 caused a death of the animals. About 50% of the mice die, whereas mice treated with BAC312 do not die, or they die just one, I think, here. Whereas similar to the control. And if you go now at this day, at day 50 after treatment, and you look at the lung and brain, you can see that the FTY720 is increasing the fungal burden in the lung, and whereas the BAF312 is similar to the control. And interestingly, if you look at the brain, you can see that a lot of mice treated with FTY720, now they show a fungal brain CFU, whereas only one mouse treated with BAF312 show a fungal brain CFU, whereas all the other ones were similar to the control. And as you can see over here, this is an histopathology of the lung granuloma. And you can see that in the control, there is a well-organized lung granuloma. BAF312 is similarly well-organized lung granuloma. But in the FTY720, we see some disorganization. And if you look very closely, you do not see these multinucleate cells. And actually, if you look very closely using a different staining called music carmine, you can see that there are a lot of cryptococcal cells outside the granuloma in the surrounding tissue, things that we do not see with mice treated with BAF312 or with control. And if you look at the primary infection model in which mice are infected with our wild type strain, we treated first, we started the treatment for 2 days prior to the cryptococcal infection with our wild type strain. And in this case, if mice are treated with FTY720 compared to the control, we do not see a very difference in survival. But very interestingly, we found that if we treat mice with BAF312, we actually see an increased survival. And it was surprising for us because we didn't know, we couldn't explain this. But then when we went back in vitro, we found that FTY720 actually is an antifungal, is able to kill fungi. This is an MIC of FTY720. And as you can see that FTY720 is able to kill Cryptococcus wild type, Cryptococcus mutant, Cryptococcus gattii, which is a different species, is also able to kill Candida albicans at 4 microgram per ml, whereas the phosphorylated form is not. And that's why we think that FTY720 did not have any effect here because it's rapidly phosphorylated by the mouse, and therefore it does not induce any protection. Whereas the unphosphorylated is really active. Now, BAF312 is basically acting similar to the unphosphorylated form of FTY720. And we know that FTY720 is phosphorylated only by the host J774 mammalian cells, and FTY720 cannot be phosphorylated by Cryptococcus. The SK1 or SK2 or crypto does not phosphorylate FTY720, suggesting that this lack of effect is due to the host phosphorylation. And as you can see over here, when we look at the CFU, you can see that the brain treated the brain of mice treated with BAF312. They have some of them, they have some CFU, but some of them, they do not have CFU, suggesting that some of these mice actually were able to clear the infection. Now, going back to the model, the reactivation model, we look at the immune response, particularly at the recruitment of immune cells in the blood and in the lung. These are done at -1 day, which is a day before the infection, the day before the treatment. These are -1 day before the treatment with the FTY720 or with the BAF312. To make a long story short, we could not find any difference between BAF312 and FTY720 in the recruitment of immune cells. Yes, we found that both treatment, FTY720 and BAF312, they cause a dramatic lymphopenia, CD4 and CD8, in the blood. We found that this lymphopenia was also observed in the lung tissue for both CD4 and CD8. We found some difference here in the blood neutrophil, particularly in the cell treated with the FTY720 compared to BAF312, but only at day 20 post-treatment. And also some lung neutrophils were increased, but unfortunately, this was not highly significant. Now, we look at the different cytokine produced by these cells in the lung tissue. And again, we couldn't find a difference between FTY720 and BAF312 that will explain the reactivation of Cryptococcus in FTY720 animal compared to the lack of reactivation of BAF312 treated animals. As you can see, there is some difference, obviously, in the TNF-alpha, particularly on the treated mice. But unfortunately, there is no difference between BAF312 and FTY720. And so we went back and looked at the granuloma structure. And this is where we found the major difference. As you can see here in the FTY, this is a staining of macrophage staining, F480. And this is a BVG staining in which we are staining the collagen deposition. And what is striking is that the structure of the granuloma at the interface between the multinucleate macrophages and the necrotic tissue is subverted. So in the control in BAF312, you can see that the multinucleate cells containing Cryptococcus are inside the granuloma, surrounded by this fibrotic tissue. And this is similar to the BAF312. But in the FTY720, you see that a lot of multinucleate macrophages containing crypto is actually outside. And this is confirmed using the VDG staining. And if you do a calculation of this F480 inside versus outside granuloma, you can see that the FTY720 has a significant difference compared to the BAF312. So clearly there is an effect of FTY720 in disorganize the granuloma structure. And so this is also present in if you do an M1 and M2 labeling at the granuloma site. And as you can see, if you do the calculation between CD38 and ERK2, you can see that the FTY720 is clearly disrupting this ratio at the granuloma site compared to the control in BAF312, the treated animal. And so we look at the possibility of which sphingosine-1 phosphate receptor will be important for this effect. And we all know that the fingolimod is targeting S1P receptor 1, 3, 4, and 5, whereas siponimod is not targeting S1P receptor 3. And so we focus on S1P receptor 3. And what we found was really exciting because we did a pharmacological treatment first with FTY720 phosphorylated and different inhibitors. And as you can see, inhibitors of SIM 5541, which is actually in specific agonist of S1P receptor 3, is actually stimulating the phagocytic index, whereas all the other one is blocking the phagocytic index. And phagocytic index is the ability of macrophages to internalize Cryptococcus neoformans. And this internalization is important for the killing of the organism. And indeed, if now we are treating, which is normally through a ROS production, and if in our system, if we are treating with FTY720 phosphorylated form, we are seeing that the ROS production is slightly decreased. compared to the treatment with other compounds, particularly BAF312. And what was more interesting is that we corroborated this effect on the S1P receptor knockout animals. And if we take the S1P receptor knockout animals, we see that the phagocytic index is slightly decreased. And this is also not restored by the supplementation of a low amount of S1P. Suggesting that this phagocytic defect is really mediated by S1P, through S1PR3. And as you can see over down here, the killing, not only the phagocytic index, but also the killing of the fungus through the macrophages appear to be mediated by this effect of S1PR3, because the killing is decreasing in the S1PR3 knockout macrophages, and not restore upon the supplementation of S1P or different doses of S1P. It suggests that both the phagocytic index and the killing of the organism appear to be mediated by S1P through S1PR3. And because of this, ROS production is also slightly decreased in the S1PR3 compared to S1PR3 mutant wild type. And so therefore what we think is that the S1PR3 through S1P, through S1PR3 is controlling the macrophage response at the granuloma sites. And the model of our infection here is that so if S1PR3 is important for this controlling of the granuloma formation and function of the macrophages at the granuloma sites, when FTY720 is coming, it's basically binding to S1PR3. And S1PR3 is no longer available for S1P to do whatever it needs to do at the granuloma site of crypto. Whereas BAF312 is totally fine because it doesn't target S1PR3, and therefore is not reactivating. Therefore, S1P can bind to S1PR3 of these macrophages, and therefore protect the host from reactivation of the granuloma. And so, what I did show you is that the multiple sclerosis patient taking fingolimod are more susceptible to cryptococcosis, fingolimod but not BAF312, reactivated cryptococcosis from the lung granuloma. Fingolimod or BAF312 cause significant lymphopenia in blood and in tissue. And this is important because, I mean, even though the lymphopenia is basically identical, only fingolimod reactivated, but not BAF312, which is also interesting because it clearly suggests that the cryptococcosis is a disease in which lymphopenia is necessary but not sufficient for crypto to come back. And this is also important because that's why I think not every single HIV patients get cryptococcosis. It's not as frequent as one may expect. Something else must happen. And I think this something else has to be with the granuloma, particularly with the granuloma structure. And again, I show you that fingolimod impairs cryptococcal phagocytosis killing through S1PR3, a receptor that is not targeted by BAF312. And I thank you for your attention. This work against was now is taken over by Tyler, but it was done by Karen and by Ariel, which is not taken in this picture. This is just a recent picture. And this is my group. And I thank you very much for your attention.